Okay, so good morning. Thank you for joining us to the NewTechAqua webinar series. Today we have a new webinar about the circular aquafeed ingredients for sustainable aquaculture. Before we start to the webinar, I would like to make a brief introduction of NewTechAqua project. Just to let you know, to be familiar with the main objectives of the project and the research that this important project has implemented during the 4 years of duration of the project that is finishing this December 2023. So the project was approved under a call launched in 2018 and 2019 about the sustainable European aquaculture 4.0 nutrition and breeding. Under this important call, 4 projects were approved. Those are the e-efficiency, intelligent fish feeding through integration of enabled technologies and circular principle, aqua impact, genomic and nutritional innovations for genetically superior farm fish to improve efficiency in European aquaculture, NewTechAqua, our project, new technologies, tools, and strategies for a sustainable, resilient, and innovative European aquaculture, and FuturEUAqua, future growth in sustainable, resilient, and climate-friendly, organic, and conventional European aquaculture. NewTechAqua is innovation action. It means research focusing on innovative solutions and tools for developing a sustainable aquaculture. The project is based on a consortium of 25 partners. 11 of them are coming from research and development institutions. The project is coordinated by University of Bologna, and there are partners from Italy, University of Venice, IRTA, HSMR, UNIBO, NOFIMA, IFREMER, CISAF, DFMR, the University of Las Palmas of Gran Canaria, and CNRS. And there are 12 partners coming from the industry. Some from, there are fish farming companies, other aquafeed producers, biotechnology companies, and 3 partners that are, let's say, working on exploitation and dissemination. Those are the FEAP, the Federation of European Aquaculture Producers, CIHEAM, the International Center for Advanced Mediterranean Agronomic Studies, and Green Innovate. So the objective of the project has been to expand and diversify aquaculture production of fin fish, mollusks, and microalgae by developing and validating technological, advanced, resilient, and sustainable application. NewTechAqua has focused its work on the most important fish species nowadays produced in Europe. Those are salmon, trout, sea bass, and sea bream. But has also worked on the development, optimization of the reproduction of emerging new species as amberjack, migre, sole, and mullet. It has also worked on mollusks, oysters, and mussels, and with 3 microalgal species. The project has some multidisciplinary and has worked on information technologies, on development of feeds and feed strategies, on farming production technologies, development of new products, optimization of the reproduction of new species, and also genetics and breeding. Also, the NewTechAqua has made a great effort for the dissemination of the knowledge generated by the project, and made it through what we have done. We have called it a training program. Under this training program, we have organized 2 advanced courses, a course that we did organize in Zaragoza about nutrition and feeding, a second course that is organized in Tricasi by our colleagues from CIHEAM Bari about organic aquaculture, 2 workshops, one workshop that was organized in Spain about the low impact aquaculture systems, and a second workshop that is going to be organized in Italy, in Venice, about precision farming. And then we are developing e-learning modules. We are organizing a very important webinar series. Today is the webinar that you are participating about circular aquafeeds. And then we are finally implementing a school campaign. This is the program, as I told you, very intense for webinars. Today we are organizing the second webinar during the month of October about circular aquafeed ingredients for sustainable aquaculture. You have there the list of the webinars that we are implementing this month, but we have organized 3 webinars before. In case that you are not able to follow us on live in these events, you can always watch the past webinars on YouTube. And just to let you know that next November, 15th of November, NewTechAqua is organizing its final event in Brussels. Just a practical issue for you to be familiar. We are using Zoom, and in Zoom you have this question and answer tool. That is the one that we are going to use today for you to help you to make your questions to the panelists of speakers that we have today. And there is also a chat, but this chat is not working well for the webinar. So just for the questions, work with the questions and answer tool. We will be watching your questions, and then we will try to answer that. Luca, if you would like, please, to continue.
Yes.
Mm-hmm.
Good morning, everyone. My name is Luca Parma. I work at the University of Bologna as a fish nutritionist, and I will present briefly and will introduce the work package 1 of the NewTechAqua project. Work package 1 is focused on the development of feed and feeding strategies with several objectives. The first was to improve the fish health, reducing spiricot increase of infection in Cerri using natural functional diets. Then in this work package, we promote marine byproduct valorization, employing innovative free catch fish meal diet in Cerri. That will be the topic of today's webinars. We also have developed organic low fish meal aquafeed in conventional and emerging species. This was presented the 3rd of October in this webinar series. And also we apply some microbiome application to enhance health of farm species in relation to feeding regime. In the work package 1, there was a strict collaboration among research institute, University of Bologna and IRTA, together with feed industry and farmer, Veronesi, Cromaris, Irida, and Galassidi Marine Park. Just a brief overlook of the task. All the methodologies that we employ in this work package was based on the development at the laboratory scale of the feed that we tested, such as in the first task, the ProHealth feed against spiricot yield. And then we made a validation of the feed tested in real cases. The similar approach we did for the development of innovative, sustainable feed for European sea bass that we tested lab scale, and then a validation, the results were validated in Cromaris in this case. The organic feed were developed for trout, sea bream, and meager by the IRTA in Spain. Also, with the main scope of this task was to find novel alternative vegetable organic raw material that have been tested to reduce fish meal in these species, and again with a strict collaboration with the farm, in this case in Galaxy Marine Farm, we tested in real cases. Today it will be the focus on circular aquafeed ingredient. That is, I think, a very hot topic for the sustainability of aquaculture. Now the big problem for all the aquaculture is to become more sustainable, to find sustainable alternative raw material that are necessary to continue the growth of our sector. And the program will be mostly the presentation of the NewTechAqua result, but we have invited, and I would like to thank you, Elena Mente, also from University of Thessaloniki, that will present result from FuturEUAqua project, a sister project of NewTechAqua. Basically, she will start and she will present organic fish feed for the futures. After that, Roberto Cerri from Veronesi will present the role of circular ingredient for aquafeed industry from the point of view of the industry. Myself, I will present the result that we performed at the University of Bologna in lab scale experiment of on the use of alternative byproduct from fishery and aquaculture in European sea bass. And follow my presentation, Silvia Krisianak from Cromaris, she will present the result that we achieve on the testing of the diet that we develop a lab scale trial in real farming condition. I would like to thank all the lecturers of this webinar and all the participants. And I hopefully will have a fruitful discussion at the end. I remind you that Elena Mente will not be with us until the end of the presentation. So if you have any question after her presentation, just we can make a very quick brief discussion. So thank you, and I would like to introduce Elena for our talk. Thank you very much.
So I'm going to share the screen now. Thank you, Luca, for the invitation to participate in this webinar organized by NewTechAqua. Okay, I think it's going too far. Right. So I'm going to talk about the organic fish feeds of the future, and I'm going to present you some results from the FuturEUAqua project. As Luca already said, I am Elena Mente, and I'm a professor of aquatic animal nutrition and physiology at the Aristotele University of Thessaloniki at the Department of Veterinary Medicine. My aim today is to present and point out the relationship between the innovative fish feeds and nutrition to produce a healthy fish, and also to understand the role of nutritional research in sustainable and organic aquaculture. So it is well known that aquaculture is the future of food. In 1968, we know that the contribution of capture of fisheries versus aquaculture to human consumption was 94% and 6% respectively, and now in 2018, the contribution of capture fisheries versus aquaculture to human consumption is 48%, as you can see in this graph. And obviously aquaculture is 52%. Aquaculture will contribute by 59% in 2030, and This means 109 metric tons of aquaculture will provide fish for human consumption, but we need 40 million tons of aquafeeds for this. So how we are going to address this problem? A lot of views are centered around European countries on issues on sustainability, eco-labeled products, food security and organic. Sustainability in the sense of sustainable use of natural resources, escaping the poverty trap, and sustainable development. To meet the needs of all these without compromising the ability of future generations to meet their needs. And under this umbrella, sustainable aquaculture best practices and innovation are taking into account the environmental sustainability and the organic aquaculture production. So we aim to increase aquaculture production, but we have to do it taking into account the environmental sustainability and obviously not forget the food security and nutritional strategies, and promote basically practices, sustainable practices, and one of these is the organic production. The organic production is a method and has a dual societal role. In one hand, provides for a specific market responding to consumer demand organic products, and on the other hand, delivers public goods contributing to the protection of the environment and animal welfare, as well as to the rural development. With the European Green Deal, the Farm to Fork strategy, and the biodiversity strategy, the EU has set targets of reaching at least 25% of the EU's agricultural land under organic farming by 2030, and this means also a significant increase in organic aquaculture at the moment. At the moment, as we see, some countries have reached already the target in general for the organic farming in agriculture too, and some countries need to aim more for organic farming and apply for this target. So the results from the FuturEUAqua project, which investigated in work package 2 the use of efficient alternative feed ingredient sources, for example, by using microalgae, bacterial protein, yeast, insect meal, fermented soya bean, and tunicate meal, and also investigated ingredient sources that can be sustainably produced with an ecological footprint. As a general principle in nutrition, I have to remind you that feed should meet all nutritional requirements of the organism, promote animals' well-being, health and growth, and ensure high quality of the final product with a low environmental impact. And when we talk about fish nutrition, there are different areas and people see differently in relation to who they are. So for feed producers, the formulation of tailor-made, species-specific, low-cost, environmentally friendly diets is what they are aiming for. Farmers, they want the optimization of feeding to promote the best fish growth. And researchers and students, they evaluate the long-term effects of diets, and nowadays novel diets, to meet fish nutritional requirements and physiological functions and obtain the best fish growth health performance. FuturEUAqua results in relation to aquafeed sourcing show that the most acceptable source of ingredients for fish feed is microalgae, and this was questionnaire that was conducted in several EU countries. Insects and GMO-free vegetable proteins follow at second place, and then we had wild fish and vegetable oil as it is listed by the consumers. So fish trimmings and bacterial proteins are right on the scales, midpoint. However, the way participants responded on the acceptability of feed ingredients varies between European countries. So FuturEUAqua developed feed formulations, as we said earlier, and the use of efficient alternative feed ingredient sources. Mentioning again that they have to be sustainably produced with an ecological footprint, but also we need to have a better understanding of the molecular mechanisms, of the control of the nutrient utilization that will help us to generate sustainable and functional diets and improve the efficiency of aquaculture in the future. So studies in the project were focusing on protein, lipid resources, responses to dietary stimulations, and information of the relationships between diet composition, protein, lipid metabolism, and nutrient utilization in aquatic animals. The plan was similar to NewTechAqua. There were experiments conducted in the lab, in different labs according to the species. So we had for sea bream and sea bass experiments conducted in Greece, in HCMR and University of Thessaloniki, and for trout and salmon, we have experiments conducted in the lab of DTU and in FEMA. Then the results were validated in a field study in farms for European sea bass and sea bream in Greece for Galaxidi and Kefalonia, organic farms, and the diets were produced by Irida. And for rainbow trout, we had Aqua produced the organic diets, and for salmon, we had Nofima, and the companies, the farms that were validated the results were Mole for trout and Samar for salmon. So let's start with some results for sea bass. For sea bass trials, we had 3 protein sources, and we selected the conventional fish meal, fish oil, 3 mix. We had novel ingredients, and to be more specific, we include a mixture of bacterial protein, which is a single cell protein with a natural fermentation process similar to making yeast. We had a yeast meal, which is a yeast-based product formed from the collection of the cytoplasmic contents of the yeast cell Saccharomyces cerevisiae. An algae meal, which is long-chain omega-3 rich algae ingredient from the native algae Schizostridrium species, and is composed of a multiple of unicellular microalgae cells. The diets were balanced to have the same protein and fat content, 46% and 20% respectively. And the results show that the novel ingredients, as we identified them before, this mixture, as was said, at moderate inclusion levels, either with conventional fish meal, fish oil, or trimmings of fish meal, fish oil, they have positive effects on growth performance and feed conversion ratio. The negative effects of the novel ingredients on growth performance evidence in this study that there are some issues when we use novel ingredients, and these issues are on palatability and on digestibility. We had a final fat accumulation in intestine and liver, but this was due to the increased feed intake and final weight that we observed. And basically, as you see these triangles, this is the results. This is just a schematic diagram, and is showing the design of the study, which is a mixture design. And in the middle of it, you could see the moderate inclusion of the 2 other protein sources that they are in the other side of the triangle. Here. Red dot in the middle is equal contribution of 4 protein sources. So we are inside here, here. And the other 3 dots inside the triangle are the diets with different levels of inclusion for each protein source. The red areas indicate a higher value record for each parameter, and the blue areas a lower while the yellow and green is an intermediate value. Overall fish fed, as I said before, the diet with moderate inclusion of conventional fish meal, fish oil, and novel ingredients as this mixture, as I described it before. They had better food conversion ratio, significantly higher feed consumption, and growth rates. High inclusion of novel ingredients, lower feed intake. If we go to salmon results, we had again a set of novel ingredients here. And these were some insect meal that replaced fish meal, tunicate meal that replaced fish meal. We had a diet with zero fish meal, zero fish oil. We had an organic fish meal, fish oil diet, which means a very high quality of fish meal and fish oil. And we have an algae diet. The results show that high protein levels in the fillet increase fillet and skin levels in the microalgae groups, and reduce cholesterol and protein in blood serum in the diet. Increased serum glucose was all with the innovative ingredients combined, and there were no significant differences in specific growth rate, food conversion ratio between the diets. And sorry, I didn't mention before, I was saying that high protein levels in the fillet increased when we had the skin zinc levels higher in the microalgae groups. So high performance, high FCR, superior digestibility when they fed the organic fish meal and fish oil, and some lower results when we used the tunicate meal. Decreased expression of stress-related genes and increased expression of lipid and steroid metabolism when we have salmon-fed microalgae and insect meal combined. So the take-home message that overall the best diet that had the very nice and organic and high quality of fish meal, fish oil, showed obviously the best results, but we have seen some similar results when we were replacing part of that with the novel ingredients like a combination of insect, tunicate, and microalgae for salmon. For sea bream, we had a low FIFO diet for organic diet. So the control diet was designed to simulate the composition of a commercial diet incorporating organic fish meal trimmings this time, organic fish oil and soybean meal, organic and concentrate. This low FIFO mixture replaced fish meal at different inclusion levels, 20, 25, and 30%. And we see that the low FIFO mixture that is consisted of organic processing fish trimmings, bacterial protein, and yeast protein at a percentage of 25% gave the best results, higher growth performance compared to the control, and very nice structural histological analysis of the liver and in the gut. We have a very good distribution of the goblet cells. For sea bream, we had a different set of diets. We had a replacement of fish meal with algae, insect meal, and tunicate meal. And we decided to have the replacement with 68% or 45%, either with the tunicate meal or the insect meal. And the algae was no fish meal at all. So we saw that the total replacement of fish meal with algae meal, insect meal, and tunicate meal, so a combination of these three novel ingredients, didn't affect sea bream growth performance compared with the 68% replacement of fish meal with insect and 45% replacement of fish meal with tunicate. In addition, we see that there is no difference between the two commercial diets for sea bream, and FuturEUAqua organic diet modify fish metabolome in a different way, as you can see here. It's very clear. Future organic and future also, because we used a commercial conventional diet too, induces different modifications in the metabolic pattern than the commercial organic and conventional diet that the industry is using at the moment. Further research on gut microbiota was trying to answer some questions like, do gut bacterial communities exhibit temporal shifts and diversity, and are these relating to the temporal variation in food supply of nutrients? What are these gut bacterial communities that could serve as providers of essential nutrients? And we know that fish cannot synthesize several essential nutrients required for their metabolism and growth and depend on the feed for their supply. Certain animal groups can use these nutrient-deficient diets as they bear symbiotic microorganisms that can provide these compounds. So diet is a major factor driving the composition and the metabolism of the gut microbiota. But gut microbiota is actively involved in a nutrient assimilation and immunity of the host organism. So can we find these compounds or microorganisms that can really synthesize some nutrients that they are deficient in the diet? So that's the big question. We realized from the test we did and from the diets that we used that compared to the control feed, tunicate and insect didn't alter the dominant mid-gut bacterial group, while 6.6% of the share OTUs of the microorganisms in the gut is very low, and this shows that the tunicate, especially diet, had a distinct microbiota. Tunicate was distinct and had also less than 1/3 of its OTUs unique and the highest OTUs recent. So we have to go back and see how tunicate could be incorporated in the diets for salmon and sea bream according to our results. The three treatments here with the algae shared only 6% of their OTU, suggesting that each one had again a distinct microbiota, and we still are searching and analyzing the gut bacterial communities that could serve and provide these essential nutrients to the fish. So we found some bacterial taxa that they had beneficial metabolic services to their host, and especially when we compared the diets according to the novel ingredients that we used for salmon and sea bream, we had some similarities in some bacteria and in some others we didn't. So innovative, species-specific, nutritionally adequate, tailor-made, and low ecological footprint fish diets, for example, the tunicate is a novel ingredient that could be introduced in this area, and their nutritional impact of farm fish growth, performance, health, and quality for a better performing, sustainable, and organic aquaculture is our future. Thank you.
Okay, thank you very much, Elena. Well, and congratulations for the big work you have done with FuturEUAqua. Very complete and very interesting. Many the new diets, alternative ingredients you have tested. I don't know if Luca or any one of our panelists would like to make a question to Elena.
Congratulations, Elena, for your work, of course. And among this ingredient, what is your point of view concerning the availability, for example, of this raw material for the future? For example, for insect meal, how do you see we can solve the availability of this raw material for the industrial level?
Yes, that's a very nice question. Thank you, Luca. The availability is always an issue, and the cost is a second problem that we face at the moment, and especially for the future. My answer will be according to the results of the FuturEUAqua project, that there is no ingredient that could be used directly and could be the best one. So it's a kind of always a mixture of different ingredients, novel ingredients that should be tested, and is so species specific that also age related according to the farm of the fish that we study. So the answer is that still we need to research more on this. I could call it cocktail of novel ingredients per species, and also improve sometimes some new ecological ingredients like tunicate that they need more work on how to be applied and be available and processed better in a way that the fish could digest it easier. But yes, the future is there, so definitely we can use these ingredients, but in the right quantities and in the right mixture. We have to find the cocktails.
Yeah, and also continuing with this is I see that the work you have presented, these novel ingredients that in the short term are not going to be novel. They are going to be incorporated in the offer of available raw materials. Is that what I see is in principle there are they are not ingredients, single ingredients, they are more a group of ingredients, no? Because when we are talking about microalgae, it's a new world that we are opening. Insect fish meal, I'm sure that there are differences between the origin and the species of the insects we are using. I have a question about the fish trimmings, because it seems that according to your results and other results, they have a lower palatability and lower digestibility. I know that they can be incorporated, but how do you see this question of lower digestibility or palatability compared with other alternative ingredients?
Thanks for the question. The issue with fish trimmings is that definitely we have to use them because it is a waste not to. But this lower palatability usually is because of the high phosphorus content that they have. So we need to further process it and try to find a way to reduce this phosphorus content, or we need to go back to the mixture, as I said before, and find the right quantity that we have to incorporate them in the diets when we use per fish species, as I said before. So yeah, it is for trout. It worked a little bit better, but again, when we added more ingredients and we reduced it. So still, yes, we can use it, but not as it is.
Well, but the good thing is that you are proposing new lines of research to overcome those problems. Okay, thank you. Well, we should thank you very much, Elena. You should leave. Thank you for joining us today. We have to continue with the webinar. So cheers, Elena.
Thank you, and nice to see you all.
Thank you.
Okay. So we continue with Roberto Cerri from Veronesi, who is going to make a presentation about the role of circular ingredients for the aquafeed industry. So Roberto, please.
Okay, thank you, Bernardo. Okay. So good morning. I'm Roberto Cerri from Veronesi, from AIA. And today I'm going to show you a presentation about the role of the circular ingredients for the aquafeed industry. As I said before, I'm Roberto Cerri. I'm a marine biologist. I'm a product manager in Veronesi. Here you show my contact, my email, my mobile phone, so you can contact me if you want. Veronesi is a big company, is the key role in play a key role in the production of animal feed and meat sector. Veronesi is a premier brand of a group. It's the largest feed producer in Italy, more or less 3.2 million of ton of feed produced in 7 plants. more or less 40,000 tons of aquafeed produced just in 1 plant. As other Italian and European group, Veronesi is constantly in contact and work with Assalzo and FIFAC. Okay, what does circular feed mean? The concept of the circular feed is to recover the recovery of the nutrient and the reduction of the nutrient losses. Products derived from the livestock farming that are an excellent source of nutrients, and this product can be consumed by human and animal. Of course, the quality of these two products for the human and animal consumption showed a different quality. So in this slide, I will explain a little bit the legal definition of the co-product and byproduct. So the difference between them is the economic value or the volume obtained from the main raw material. Byproduct is not a waste with a negative meaning. It's a co-product with still high nutritive value. A co-product is recognized for its nutritive value and treated as the main product, so as the raw material, the original raw material, maintaining its quality and its safety characteristics. A greater socio-economic value must be recognized, for example, for the reduction of the carbon footprint. Otherwise, its use is discouraged. This is present, all these information are present in the FIFAC document that you see here on the right. I'm going to present you some example of the raw materials and then the co-product. All these examples are present in the Feed Sustainability Charter 2030, published by FIFAC. So in this slide, you show the raw materials, so soybean, rapeseed, and sunflower seeds that are cultivated to produce vegetable oil and biodiesel. But there are 3 co-products that are soybean meal, rapeseed meal, and sunflower meal that are so interested for the feed, animal feed. The other slide about the corn. So the corn is cultivated for the starch, mainly for the starch is the main product obtained, but we have 2 co-products that are the corn gluten meal and then the dried distillers grain pellets. The other example is the animal, the carcasses of the animal that are ready for the meat, but we have some byproducts, some co-products that are processed animal protein, animal fat, and spray-dried blood plasma. Another one is the microorganisms grown in industrial fermenter that are cultivated for the food and feed additives like amino acids. we obtain a biomass, a microbial biomass that is so interesting. So in this here, we are reading a lot of paper with single cell protein. So because the content of protein is really, really high. And then other two ingredients, co-product or byproduct. The fish trimmings from the head, the backbones, and the viscera of the fish, and then the hydrolyzed feather meal that are a co-product from the poultry processing operation. Also, this co-product is high in protein. So far, we have seen the general recommendation of the FEFAC. Regarding aquaculture, the guidelines and the recommendation for the circularity of the fish feed were published by the European Union in July 2023. So the circularity in feed production is a concept that is still in process of being defined. A provisional definition of the circular feed could be not food grade ingredients recovered as secondary raw material from the local circular economy with a small land use footprint. So we need to consider that when we are evaluating our raw material, so the food feed grade status, the proximity of the origin of the feed meal, the land use ratio, the forage fish dependency ratio, and also the nutrient digestibility. In the NewTechAqua, we did 3 trials, 2 in the lab and 1 in a farm. We tested 12 diets, and 2 private companies were involved in the project, and also 2 consortium. What we did take into consideration, so the forage fish dependency ratio, that indicates the amount of wild fish used to produce 1 kilogram of fish in the farm. And this value was calculated according to the ASC farm standards. I need to tell you that trimming fish meal and fish oil are not included in this calculation, just wild catch fish meal and fish oil. In this slide, I present some ingredients, some co-products that we use in these trials. So shrimp meal, fish oil, vegetables, and innovative ingredients. So fish oil and fish meal that we formulated the test diets, replacing wild catch fish meal and fish oil with the trimming fish meal and fish oil. The production of the fish meal and fish oil from trimmings is exactly the same of the fish meal produced by wild catch. The only difference is the origin, fishing vessel or by catch or by product. Sorry. About the shrimp meal, we included the shrimp meal obtained from the dehydration of the raw material. The production, the process of production is extremely important to preserve the quality of the protein, the pigmentants, and also the digestibility of this raw material. About the vegetables, gluten as a vegetable proteins. On the right, you see the amount of gluten obtained from each raw material, with the highest level in wheat and the lowest level in legumes. And then the innovative ingredients. In these trials, we use macroalgae biomass produced in Almeria in ponds. All the raw materials should be evaluated in terms of availability, nutritional quality, and sustainability. Of course, the availability is so important because if we want to introduce an ingredient in a diet, we need to be sure that there is a sufficient availability of the product, and the nutritional quality. For example, here I show you the amino acid profile of the corn gluten and wheat gluten compared to the amino acid profile of the fish meal, and we see that there are some lack of some amino acids like lysines, glycines, and arginine. About the sustainability, we see that in the graphs on the left, the amount of co-product used in the last 20 years increased a lot. But we need to consider that in the future, the European Union put a brake on the import of products like soya from the deforestation area. So maybe the quantity of the co-product from the soya maybe is a little bit difficult to find this product. And in Europe, how we can evaluate the sustainability of an ingredients with the use of GFLI, so the Global Matrix for Sustainable Feeds. The GFLI partners are developing a database and tool to calculate the LCA. Here I show you some example about the kilogram of CO2 equivalent produced in aquaculture. So, for example, for 1 kilo of fish, it's more or less 5 kilo of CO2 equivalents. 1 kilo of fish feed between 1 or 2 kilos. 1 year of Mediterranean diet, 1,087 kilogram of CO2 equivalent. 1 year of USA diet, bit lower, the value is higher, 3,300 kilo. A vegan diet, 1,500. And other example about the driving a family car for 1 year, more or less 5,000 kilometers, with electric car, 5,400 kilos, and biodiesel, 7,700 kilos. And also an example about the passenger traveling from Frankfurt to Ibiza, more or less 4 people, 2,096 kilo of CO2 equivalents. Okay, after this presentation, the solution for the sustainability and the circular economy of the aquafeed is a zero waste diet for sea bass. This is the product that we produced. Thank you so much.
Okay, thank you very much, Roberto. So we are continuing with the presentation. Please stop sharing the screen. I do remember the attendees that they are able to write their questions, doubts in the questions and answer tool. So please use it. We continue with today's webinar with Luca Parma presentation from UNIBO to presentation called, well, you make it better than me, Luca.
Yeah, thank you very much, Bernardo, and thank you again, thank you, Roberto, also for your very focused, industrial focused presentation. I'm going to present the result that we produce within the NewTechAqua project in collaboration with Veronesi and with Cromaris towards a free wild caught fish meal, fish oil, and soy protein European sea bass using byproduct from fishery and aquaculture. Just a briefly introduction of myself. I'm a senior researcher at the Department of Veterinary Medical Sciences, University of Bologna. I work mostly on fish nutrition, on marine fish species, and mostly I've been working on protein requirement, on alternative protein, and also on gut health functionalities. So the major outcome will be the result of, as I said, of the NewTechAqua project concerning the trial that we perform in our laboratory and at the University of Bologna. So just a brief introduction. In the last 20-30 years, we made a lot of effort to replace marine product, fish meal and fish oil, mostly using vegetable ingredient. Soy bean meal, for example, was a big issue in the last 20 years. Now the challenge, as we have introduced, is to work towards circularity, sustainability, so adding ingredient that are coming and are produced through a bioeconomy approach. These ingredients, as we said, are mostly recognized on single-cell proteins, some of these, insect meal, and also byproduct from fishery and aquaculture. The interest in these ingredients are the composition, the high protein content, which are also the quality of the protein is usually well balanced. The possibility of this ingredient that are also to give a lower carbon footprint on the feed, which is considered on the most important impact also in the aquaculture sector. These ingredients are also a source, maybe also a source of a functional component. This has to be taken into account. The single-cell protein are known to contain nucleic acid, for example, insect meal, chitin, lauric acid, and byproduct, when they are treated with hydrolyzation, are also sources of bioactive peptide. Fish byproducts are well known. Fish meal by trimming and fish oil and trimming are already well used and already represent mostly about 30% of the fish meal which is used worldwide. Still a lot of possibilities around this ingredient to increase the utilization. They are known to be rich in macro and micronutrient, and they can also reduce, of course, fish in and fish out ratio and reduce the GHG emission in our production. Has been also recently studied the impact of the byproduct from fishery and aquaculture, which is even lower than some vegetable ingredients such as soybean. So the aim was exactly that, to replace the wild caught fish meal and fish oil and soy protein using a byproduct from fishery and aquaculture and other alternative protein for the growth of gifted sea bream. We conducted a first trial where we aim to replace fish meal prime and fish oil prime using fish meal and fish oil from trimming, and it was a gradual replace, and also we aim to substitute soybean meal using glutens. And also we did include some, say, innovative compounds such as yeast and microalgae sources. The aim was a gradual replace of all these ingredients to have a total absence of wild caught fish meal, fish oil, and soy protein. The diet were isoprotein and isoenergetic. Were tested in the facilities of the University of Bologna in Cesenatico, with a standard procedure for the feeding trial that we usually perform. It is visual satiation, treatments, standard optimal temperature for European sea bass, and optimal environmental condition. We started with an initial weight of 75 grams. At the end of the trial, we measure growth, body composition, somatometric index, but also we evaluated parameters such as plasma biochemistry to assess the health and the nutritional status of the animal. The gut health was also performed by the study of the gut microbiome by using Illumina next generation sequencing platform that we collected the data from distal intestine content. Going to the result of this trial, at the end of the trial, all the fish tripled their weight according to standard nutritional trial. And we observe no differences when we change the fish meal by trimming with the fish meal prime using fish meal by trimming, as we can see here. We saw slight differences in growth and final body weight when we totally replace both fish meal and wild fish oil. In the growth, we had at the end slight differences. In feed intake, we did not observe differences for the acceptability of the animal when we totally replace with the say zero waste diet in comparison to a control diet, as well as the FCR was not affected in the total replacement in comparison to the control diet. We had an higher FCR at an intermediate replacement, but where we also did not include the innovative, the adding of the innovative ingredients such as yeast and microalgae sources. Similar also, the protein efficiency ratio and the gross protein efficiency were generally let's say, well balanced between the diet, with again just smaller differences in the intermediated replacement, but not in the total replacement. And also the biometric indexes, we observe a general increasing of the hepatosomatic index at the end in the total replacement compared to the control diet. This could be related to the inclusion, for example, of the gluten's. We also measure the muscle metabolomics that we saw some increase of amino acids, some glycine that could indicate differences, for example, in protein catabolism. And it was also quite interesting to see differences, for example, in the creatine. That's in the taurine. This could be an issue when we replace wild fish meal from using byproduct from fish and aquaculture, using fish meal by trimming, since creatine could be less present in this raw material, because it's usually well abundant in the muscle, and so also in the entire fish we produce for the fish meal. The plasma biochemistry test in general is standard parameters that were allocated within the welfare condition of the animal, within the range of optimal welfare condition. The most interesting, significant that has to be evaluated was also related to the creatine and uric acid, which was diet, the control diet. It could be also related to the metabolic activity in relation to the protein digestibility, metabolism, protein metabolism. The gut microbiome, we did not observe a shift among the different composition of the gut microbiome, which was similar. So we did not get a clear impact of the diet on the overall structure of the gut microbiome, as we can see in these unweighted unifractal distances that were not separated, the cluster, as well as the internal biodiversity was not affected by the different diet. However, we saw some specific taxa that were affected. For example, we had some increase in the bacilli that was quite interesting, increasing when we replace fish meal by trimming using a byproduct from fish and aquaculture, and also some other increase in lactic acid bacilli, such as Leuconostomy. And as we can see also in this slide. So in conclusion, in the first study, we highlight the potential to totally replace wild-caught fish meal and fish oil and soy protein using byproduct from fish and aquaculture and gluten with only a marginal reduction of the overall performance. When wild-caught fish meal was replaced by fish meal byproduct, no differences were recorded but we had some differences when we combined the replacement of wild-caught fish meal and fish oil together. The replacement of soy product with more sustainable vegetable protein ingredients, such as wheat and corn gluten, did not further alter the growth of the performance, and the inclusion of a small amount of 2% of yeast in the zero wild-caught fish meal, fish oil, and soy protein was probably responsible for restoring the FCR and the protein efficiency to that of the control group. Going to the musculometabolomic, we had a suggestion that there are some changes or adaptation in the metabolic pattern for energy production at the muscular level when fish were fed fish meal and fish oil byproduct, and that's a very interesting outcome, I think, and it will deserve further investigation in concerning also to the creatine and taurine metabolism in carnivorous species such as European sea bass. For the gut microbiome, there was not a major implication in the effect of the zero waste diet on the gut health, but we had some increasing of promoting some taxa, such as Weissella, Streptococcus, Bacillus enterococcus, that would have a positive effect on gut health. I'm going to also briefly introduce, present a second trial that we perform always within this topic. Is in this case, we combine the replacement of wild-caught fish meal and fish oil using fish meal and fish oil by trimming, but in this case, we also included microalgae as sources of protein to replace soybean meal. As Roberto said before, this trial was also combined with another European project, Sabana, which we involved the participation to test the microalgae biomass that they produce within this European project. So the approach was similar than I said before, but in this case we decided to work on more juvenile stage, because we know microalgae in this case are good sources of protein, but may also contribute to fatty acids that are usually more interesting and have more requirement for the juvenile stages. So at the end, in this case, the zero waste diet was without soy protein concentrate and with a 15% inclusion of microalgae. Similar rearing condition than what I said before. The differences was the initial size that was smaller in this case, and let's say Going to the result, in this case, we did not observe any significant differences in the growth performance when we totally replace wild caught fish meal and fish oil and also soy protein. All the performances were equal, including the growth and the feed utilization. Also, this was the case for the digestibility. We did not observe any differences in the protein dry matter digestibility, as well on the nutritional indices that are related to protein and lipid efficiency. We tested in collaboration with the University of Almeria, also the digestive enzyme activity, with the major proteases were not affected by the diet, such as, for example, trypsin. We had differences in alkaline phosphatase and chymotrypsin, which increases at the certain level of microalgae inclusion. We can see in this slide better when we replace, when we introduce 50% of microalgae that corresponded to 7.5% of microalgae inclusive, we had a higher alkaline phosphatase activity, as well as higher chymotrypsin, in this case, both in anterior and in the posterior intestine. The gut microbiome, in this case, was clearly affected by the diet tested, and we saw a clear separation clear effect when the microalgae was included. So there was an effect on gut microbiome, and the mostly bacteria associated were some Clostridiaceae, but also some lactic acid bacilli that I will show you later. And also the gut microbiome diversity increase when we include the microalgae in the diet as well. Here we see some specific differences of specific taxa. We saw the increases of, for example, Lactobacilliaceae and Clostridiaceae that were also defined in previous study when we included other, for example, single-cell proteins such as bacterial protein. We had similar result that we are still analyzing for the functionality, but are promising result. So in this case, the study highlighted the possibility to totally replace wild caught fish meal, fish oil, and soy by using product from fishery and aquaculture, and adding also microalgae as a soy replacement, without affecting growth, digestibility, and nutritional performances in European sea bass. So thank you for the attention. These data are already published in aquaculture for this year, and the second trial is going to be published soon. The paper is an open access paper, so can be viewed by all. Okay, thank you very much for your attention.
Okay, thank you very much, Luca. So we continue, and now we have the last presentation for which we have today with us Silvia Krisanak from Cromaris Fish Farm Company, and she's going to talk about the zero waste feed by using marine byproducts from fishery and aquaculture, a case study. You have seen how NewTechAqua has worked in the development at the laboratory level on research for development of these new diets, then which has been developed by a aquafeed company, Veronesi, and now we are going to see the results at the farm level. So Silvia, please.
Thank you, Bernardo.
Okay.
Okay, you see?
Yes.
So I will today speak about what happened in the case study with zero waste feed. I'm a head of fish feed in Cromaris and also responsible for the feeding trials on the farm where we already did this trial with NewTechAqua. A little bit about Cromaris. So we are producers of sea bass, bream, eager, and new species, Mente and Cerriola. We have seven farms in Adriatic Sea, one hatchery, and also one processing unit in Zadar area. We are very focused on this natural farming method to have sustainability aquaculture, as we are for more than 4 years certified by ASC standards. Our sales was last year more than 30,000 tons of equivalent whole fish equivalent, and more than 100 people working in Cromaris. So about the trial we have conducted in our farm, we already have some experience to have feed without wild catch fish meal and fish oil, but we didn't have this opportunity to test products without soy protein. So that was our decision to test the absence of soy protein. That was already considered, and we had some previous result on the lab scale performed at University of Bologna. So that was the results performed in Bologna and the diets that were tested in the lab scale. Our feed was produced in Aia, in Veronesi, and our feed was what we used in the farm scale was without any soya protein, soybean meal, neither soya protein concentrate, with the inclusion of yeast and microalgae products. The test was performed in Cromaris farm at Lamiana. The control and zero waste cages, we had the duplicate condition for cages of 500 meter cubic. Started in February 2022 and ended in December 2022. Initial number of fish per cage was 10,500 tons, with the average initial weight of 100 grams of feed. Feeding method was manually every day, 4 meals per day, and the amount of feed we mostly did 7 days maximum ad libitum feeding. And in the next 6 days, it performed at 80% of that amount. Daily data that we collect on the cages was the amount of feed used per cage, temperature, and oxygen. On monthly basis, we did biometry on each cage, 100 pieces per cage. We measure length, weight, and then we did the calculation of FCR and also the condition factor for each fish. Mortality was also regularly checked 2 times per week, and we registered for this biomass calculation in the program. At the end of this growing period, fish were also sampled of morphometric parameters, plasma biochemistry, gut microbiome, and also the fillet acids composition. You can see the cages where the fish were in the tanks, and also the sampling performed at the end of the trial. So at the end of the trial, we measured the quantity and the percentage of the mortality as the survival rate of the cage, feed conversion rate, condition factor of the cage, specific growth, viscerosomatic and hepatosomatic indexes, plasma biochemistry, and fillet acid composition. All these results were analyzed statistically using the ANOVA analysis. The results performed the control diet were more or less without any significant difference between the rearing period. At the end of the trial, we noticed changes in the growth, where the control diet performed a little bit better than the zero waste feed. Compared to the mortality rate, the survival for control and the zero waste group was with no big difference. There are one cage at zero waste feed that performs lower, so it impacts a little bit in this group. The influence on FCR was the last month of the performance on growth, which affect the higher conversion feed conversion ratio in the zero waste group, which also at the end can perform the economical effect on the cage. Condition factor were without too big difference, and also the specific growth were at all time at the end the better performed in the control group. The viscerosomatic index in control and zero waste were without big changes. Higher was in control group than in zero waste, but hepatosomatic index was higher in the zero waste group comparing to the control diet. The plasma biochemistry actually showed that there was no big difference in between control and zero waste group, which show us that actually the zero waste feed it's good to use and can and also is confirming the results obtained on the lab scale. There was no big difference, but some changes in the high density cholesterol and triglycerides, which was shown here. Compared to the fatty acids profile, that also it's very important for the final quality of the product of sea bass in this case, show that the difference between those diets didn't affect on the fatty acids profile of the fish at the end of the trial. For example, here the important thing is the contribution of omega-3 and omega-6 fatty acids profile, which in these both diets were very, very similar. Also, important thing is how the different diet will influence on the sensory analysis. There are some difference in the intensity of the smell. The control diet performed like a sweeties taste of the fish, and but the overall conclusion of the sensory is that at the end the zero waste feed didn't affect on the final quality of the product and the tastiness of the fish. So the preliminary results in the farm were confirmed. The data obtained laboratory level displayed minor difference in the growth, and also we obtained the similar results on the farm scale. But at the end, we can consider that elimination of soy products can be replaced with more sustainable ingredients like microalgae, yeast, and other vegetable glutens. And that's all for me.
Thank you very much, Silvia. Thank you very much for all the
Thank you.
speakers. Okay, could you please stop sharing
Yes.
the screen so we can see much better each other? So we open now the floor for the debate. There are some questions here that have been written. Okay, so we have from Massimo Cayano a question asking about the very importance of communicating to consumers about the safety and quality, I suppose, of these new sources of raw materials. Because consumers may think or often think that, well, who knows what is in the fish feed. Maybe Roberto, you might answer or comment this question.
No, it's really important what Massimo said. Because if I think maybe portable product meal or blood meal or hemoglobin or something like this kind of ingredients or single cell protein, maybe the customers, the clients are a little bit scared about these ingredients. So it's very, very important to communicate to the customers the safety of this product, this co-product or byproduct. And but I think the things are changing because about the sustainability of the aquafeed of the farms in general. Now the customer are changing the idea. So I think yes, we need to do a lot of work to communicate all these things, but I think the things are changing a little bit.
Okay. What could be the opinion from the producer's point of view? I don't know if Silvia, you would like to comment about this question.
Yes, very important topic for us, and this communication to our consumers, it's our very hot topic, and we are in continuously communicate actually the composition of our feed to our customers to have and to be aware what we are using and how we are influenced to have the better quality of our fish, but also to decrease the impact on the environment. So it's very important also this great collaboration with feed producer to have this conjunction and to share the knowledge between us to also spread this knowledge to the consumer.
Okay, thank you. I don't know if Luca would like to say something or...
Yes, I think to add, of course, the role of the university of the education in this sense, it's very important, starting from school, for example, to communicate the safety and the high quality of farm fish that we have now in Europe, for a lot of control that are made at both industrial level from each batch of raw material and as well as at farming situation. So this is I think it's very important to promote more education starting from low level or high school or in the school. That's also within NewTechAqua, Bernardo, you did in this direction promotion. Yeah.
Okay, good. Okay, so well, we can continue with the discussion. There is one question or comment maybe by Magdi Abdelwahed from Egypt, and he's pointed out the question of prices, the very importance of the prices of the raw materials that affect the price of feeding and the price of the final products which are related with the difficulty of obtaining permanent alternative elements for feeding fish. So I would like to comment about this because this is very important. Maybe looking at the last presentation, you might have the idea that if the results of the zero waste fish were not so good, maybe we should go back to the control feeds. But the control feeds are normally based in something that is scarce and not so sustainable, and it's not so circular. So that is the need for going for these new ingredients, alternatives, of being more efficiency to promote circular economies. And that is the need for doing more research to balance this possible slight lower performance on the new feeds. On that sense, the presentation made by Luca show how by adding microalgae we can counteract some of the small effects that just feed based on zero waste, on on fish treatment might have. But I go back to the question of prices and the available sources of ingredients for elaborating aquafeeds, which is probably one of the main ingredients. And I want to point out at the fact that nowadays the price and the cost of aquafeeds are 3-4 times higher than the cost of the producing feed for pigs or for broilers. So this question of prices is very important, and the question of enlarging the availability of sustainable raw materials is extremely important to have a sustainable but also a more efficient sector. I'm sure that Roberto may comment on these questions.
Yes. So the price of the fish feed increased a lot in the last year. So in particular, this the price, the increased price is the availability of the fish meal and fish oil mainly. So we need to find alternative. There are a lot of alternatives, not just one alternative, but a mixture of different alternative ingredients. So in this way, we can reduce the availability. We can use a small part of each ingredient, so we don't need a huge availability of one ingredient. And then the price are decreasing for some alternative ingredients. If I think insect meal or single cell protein, the price is decreasing a little bit. So that's why we are including a lot of these alternative ingredients, because now the best solution for the formulation of aquafeed.
Mm-hmm. Okay. I don't know if Luca would like to comment on this, about prices, or maybe Silvia.
Yes, about fish feed prices, really from 2022 affect a lot the fish production, I think in all countries. So it's something that it's much more important day by day for us to control, to make optimization on feeding strategies also, but also to trying to obtain with this novel ingredient the same or equal or with small difference the results on nutritional parameters of fish and also the production parameters.
Mm-hmm. Okay.
Yes, if I can add just to join what you said, Silvia, the nutrition, the precision nutrition is very important and can help in counteract the price of the feed, just increasing the efficiency of from the nutritional point of view. That's, I think, nutritionist scientists can still do more, also in say in the overall environment that is changing. So we still have to do more, I think, to increase the feed efficiency from a nutritional point of view. but also, as you said, the feeding management is very important because saving, optimizing feeding operationally, that will help to increase the efficiency of the feed. And that's, I know many farmers in the Mediterranean are working on this, just increasing the efficiency of the feed from a management point of view. That is also very important to reduce the environmental impact. Yeah.
Okay. Well, I have a question for Silvia and those that participated in the design of the field trial. I was taking note, and I seen that as a feeding management, you use a manual feeding ad libitum. So I just wanted to ask you if you can clarify me why you went for the ad libitum and not for giving doses of feed. I don't know if you can tell me something about this.
Yeah, so we are using this like in a 7-day scale. So one day in this 7 day, we are doing feeding ad libitum to obtain this like maximum what the cage can perform. And but the next 6 days, we are feeding on 80% of that amount of ad libitum.
Okay,
so it's like a correction and to see what actually, because we are not performing the biometry every 7 days. So we are doing on monthly and the fish are growing. There are some mortalities. So it's like a checking point for us.
Okay,
An
thank
important
you.
thing to see. Yeah.
Okay, thank you very much for the clarification, because I have that doubt and say there is something that I haven't understood well.
Yeah. No.
Okay, thank you. Luca, there is a question for you from Emmanuel Prouvost about how do you manage the omega-3 composition in the different groups that you tested?
Yeah, we manage of course, the diet where the fatty acid composition was taken into account in the overall amount of DHA EPA that were equal to the requirement of were just over the requirement that are known for the nutritional requirement for the fish. Of course, it's a very interesting question, this one, because of course when we are reducing the amount of wild-caught fish meal and fish oil, we have to take into the consideration of the amount of fatty acid both for the consumer, for the fillet quality, but also from the nutritional point of view. Still, there are some nutritional requirements, as said by many publications in the past, but we have to take into this account. For that, there are some, for example, functional ingredient, I would say, or ingredients such as algae oil that can be interesting in just adding small amount of this ingredient can counteract the reducing level of essential fatty acid or polysaturated fatty acid. But it's our topic when we are going to reduce from marine sources, yes. But we have to work in this direction, I think.
Okay. There's one more question, but I want to go back again to the question of prices. I would like to ask something to Roberto. Maybe it's a secret, but you don't need to answer. It's about the prices for the zero waste. If they are competitive, slightly better than these controlled diets, or you have used. Maybe you are working now in a scale that the prices are not fixed, but I don't know if you can give us some light on this.
Yes, it's competitive. The price is less than the control diet, of course. The price of for example, the fish meal from the trimming fish meal is lower than, and the fish oil is lower than the fish meal and fish oil from wild catch. But I think we need to include other ingredients, other to be a competitive with the other diet. It depends on the customers with the diet that you want. With high level of fish meal, zero waste is perfect because the level of fish meal is high, and fish oil also. But yes, the price is lower than the control diet is good, but have many diets with a lot of alternatives, and this one, zero waste, is one of.
Which is good news for farmers and for buyers of the final products. One more question for you is relating with the fish meal from fish treatment byproducts, because I'm not very familiar with this. As far as I know, when you are using fish meal, you cannot use the same species to the one that you are using, and I suppose that this might limit
exactly
also the availability of raw materials. In your case, I don't know what species are you using? If they are from, I don't know, salmon, from capture fisheries products, or
no, of course we don't. We
maybe it's a secret, but
no, it's not a secret. We cannot use the same species, the fish meal from the same species that the diet are that the fish fed at this diet. We produce feed for trout, sublime sea bass mainly, and also eels and sturgeon. So, of course, we cannot use fish meal from salmonid. And that's why we use fish meal from other species.
Okay. There is this question from Marta Silva that is asking if the project has evaluated zero waste feed in trouts or salmon. I don't know if.
Yeah, no, we did not work. We did not work on salmonids in this project. The feed task was focused on Mediterranean species. For trout, we have been testing some organic feed with different, let's say, ingredient. But it was the only one trial in the organic task. But mostly we've been working on assembly in this project from feed. Yeah.
Okay. I have a question for you, Luca, coming back to the presentation, I think the one you were incorporating microalgae. And at the end of the presentation, you were explaining that you found a differences on the macrobiota profile.
Yes.
Which I suppose that doesn't mean that the change is good or bad. It's just that there are the different macrobiota that you are finding, no?
Yes. We saw, let's say, some in this direction, we still have to work on the functionality, because when you find differences in gut microbiota, of course, there are some taxa, some species that are known to be beneficial for the host from the nutritional point of view, from the immune status. But of course, we have to perform the specific functionality by, say, prediction, the functionality of microbiome that we are working on this. In other, it was very interesting because there are some ingredients that we also tested in our research, such as single cell protein that appear to have a specific effect on the gut microbiome. For example, single cell protein from bacterial that might contain nucleotides had a similar effect. In this case, also could have the presence of the cell wall of the of the microalgae could have a prebiotic effect, and we had a shift, for example, in Clostridia that we also observe in other trials that are also considered as beneficial taxa. But it's there are not many studies who did this microbiome using this kind of biomass from that was from Nanochloropsis gantana. So I think it's could be interesting to verify its prebiotic effect, that it seems it goes in this direction. Yeah.
Okay. Well, a comment that I wanted to raise up, we have said something before, is that you have seen that in NewTechAqua, which is an innovation action, when the research is done, is with the aim to arrive to the industry. This has been a very good example of how the research is done at the laboratory, the university. There are live trials in the university, and then how we go to the pilot scale production of aquafeeds, and then we go to the field trial. And I want to stress up the very importance of going to the field trials, and also that those trials are very difficult to conduct. They might look simple, but the companies need to set up, change their routine, and this is very costly for them. But I guess that for them is also a way to be closer to the knowledge, and to implement well, their experiments, or to test the things that are proposed by the research and academia. So maybe, I don't know, Silvia, you can tell us what is your experience, if it's been very difficult to work with, well, with this that is proposed at the research level, or I don't know, something to comment about this.
Yeah, of course, it's a challenge always to have trials. It's a different way of working in the production industrial scale than in the trial scale. But we are constantly having the trials. So here it's some little part what we perform with 4 cages in this NewTechAqua, but we are having the trial cages actively all over the year. So even now we have like 20 cages in this farm, small farm for testing, where we are trying always to have to obtain some better results and more knowledge with different or raw material or different feeding strategy or new species. There are always something that we need to find out how to perform better than and implement this also on the production scale. So we are always open to work on something new to have some better results also for us.
Okay, thank you, and congratulations for your work. There is one question made by Rachid Ganga. It's about the life cycle analysis, asking if we have compared the control and the zero waste diets for the carbon footprint. I suppose that maybe, I don't know if Luca, Roberto.
Yeah, I can comment.
I think Luca knows better than me this.
I can
this diet.
Yeah, I can comment this. We have not published yet that data on life cycle assessment. A partner on NewTechAqua is working on, let's say, in the development of SCA. As we know, to obtain a correct SCA, there are many parameters to be involved. For example, the proximity of the raw material location. But yeah, we had some preliminary data, and the use of trimming, it performed very well in terms of lowering the carbon footprint of the diet, as well as the reduction of soy. Because it's true that gluten has a very high intensive energy uses to produce, but soy has an overall higher impact in terms of land, water utilization, and especially if it's produced and imported from South America. So that is so at the end of the diet, zero waste diet will perform better in terms of carbon footprint analysis. But it's not let's say it's a difficult matter to be very precise. There are many factors to be taken into account in the calculation of SCA, and it also depends from which approach is considered. So it's not it's a light it's not an easy matter, but it's very important to working and to be open on this. So we will have a deliverable. A specific deliverable on this, let's say, virtual sustainability approach of this diet. Okay, but thank you for your question.
Okay. Well, I see there are not more questions. Well, we are just nearly after 12. We are on schedule. I don't know if Luca, you would like to comment something or any one of the panelists.
I would like to really thank the industrial partner, because I think in this project we really collaborated close to the industry, try to achieve results that are really applied to the sector. I think that is sometimes from scientific point of view, sometimes we test, it is right to test innovation sometimes at the basic level, but in this case it was really focused on the need of the industry, and it was really a pleasure to work with all the industrial partners that we have in this project. That was very important. Thank you.
Okay, I don't know, Roberto, Silvia, you would like to say something before we close?
I say thanks to the university, to the University of Bologna and all the universities that are partners in the project, because for us it's so important to do these trials, to be partner in this project, and yeah, just say thanks.
And Silvia?
Yes, I can also do this because with the institute, with scientists, also it's easier to work and that to perform all that. Also, they help us doing sampling and this all analysis on laboratory scale. So it was very nice to work with them and good experience.
Okay, well, with this we are going to finish today's webinar. Thank you very much for attending, coming and staying with us. And let me remind you that this webinar series continues, and then we have our next webinar, the 11th of October, where we are going to share with you the results about the functional diets tested in NewTechAqua to prevent sporadic colitis infection in seabream. We have obtained very good results, and I invite you to follow us on the next webinar. Thank you very much for your attendance. Okay. Cheers.
Thank you very much.
Thank you.
Bye-bye.
Bye.
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 Bye. Bye.
Vamos a ver.